Vanakkam. Welcome to this course on classics in neuroscience. In this series of videos, we have been looking at functional organization of neurons and dendrites. In this video, we will look at active properties of dendrites. Remember the dynamic polarization law that was given by Santiago Ramon y Cajal. We mentioned this in the previous videos. What does it state? Essentially, there is a directionality to the flow of activity within a neuron, that dendrites receive information and they pass on this information to the cell body further into the axon, which then go on to produce the action potentials. Further, the second concept was that between neurons also this directionality is something that we can encode. So, how information flows from one region of the brain to another region of the brain can also be deduced from these contacts or connections. So, these are the concepts that we discussed in previous videos. However, it was found that during antidromic conduction, for example, when a motor neuron is activated in the middle of its long axon, for example, it will produce action potential in both directions. One will go to the muscle, the other will go to the cell body. We mentioned that there will be a hesitation in the up spike because of impedance mismatch. We mentioned this previously. But if this causes an action potential, that action potential will essentially travel throughout the membrane, including the cell body, including the dendrites. So, an action potential that is caused by a dendrite will also travel in the cell body and to the dendrite. So, the dendrite will know as in will be able to identify what the response was to the input that it gave, a form of feedback. Did this cause an action potential? Because unlike axons which conduct action potentials, dendrites many times, most times conduct graded potentials. Is also something that we saw. This challenged the idea that the dendrites just receive information and then pass it on to the axon. Rather, they also receive information from the cell body as a form of feedback. This means that the law of dynamic polarization required revision. And this was also shown experimentally using both intracellular methods and extracellular recordings, suggesting that there is a need for revision of this notion, of this idea. But remember, this came about in the 1940s and the 1950s. This requirement for revision came about in the 1940s and the 1950s. When did Santiago Ramon y Cajal introduce this idea? In the 1900s and in the 1910s. Please do revisit that part of the videos where we have mentioned when did Ramon y Cajal suggest that this happens. Maybe in 1910s, for example. Also, this idea that recording from single unit responses, single unit means not necessarily intracellular, but recording in the extracellular matrix such that it was recording from a relatively small space. This came to be called as single unit recording. To how a group of neurons would act, population level processes or field potentials. This was also being explored at that time. So, there was an interest in understanding or gaining a deeper insight about how as a population the polarization dynamics worked. This was an area of research that was being pursued. And for this, it was sufficient to record using extracellular electrodes. But still, even if you say extracellular electrode, in the brain, this would mean relatively smaller electrodes. So, how big the electrode is, how the requirement of instrumentation would not change sufficiently or in a very big way, because in big axons like the squid giant axon, you can use the microelectrodes to record individual neuronal or axonal activity, but the same electrode can only be used to record population level dynamics in the brain, because these are much smaller neurons. These may be much smaller neurons. So, single spikes or single unit recordings essentially refer to action potentials in close by cells that are very close to the electrodes, and waves of activity that happen in around the cells wherever. Remember, this essentially referred to summed activity that was being measured from multiple neurons. Also, the location of the neuron with respect to the electrode plays a part, whether the electrical activity is travelling away from the electrode or toward the electrode. Remember, if you have a group of neurons, in some of these neurons electrical activity will be travelling towards the electrode, in some others it will be travelling away from the electrode. So, you do not get the fine grained details of how individual or small groups of neurons work. You are essentially recording from a whole bunch of neurons. This represents population level activity. This potential wave, that is essentially a vector. Summed means it is not merely numerical sum, also the vector sum. So, there are challenges with identifying where this information is coming from in single unit recordings. However, the major advantage was that it is much easier to get data from extracellular recordings when compared with intracellular recordings within the brain. That requires much more sophisticated instrumentation that was perhaps not available in the 1950s. So, a lot more sophistication was to happen. That is something that happened only perhaps in the 1990s, 2000s. Only at that time you had those microelectronic chips that could record really small units that can record from individual neurons from within the brain. This came to happen only in the 1990s and 2000s, perhaps even in 1980s maybe. One has to check the data, but the point is that there was ease of recording information from extracellular matrix rather than individual neurons within the brain. So, Paul Fatt was one of the first to explore this. He recorded from space around the motor neurons, and he showed that there is active impulse invasion in dendrites. For a long time it was thought that it was only the axon that was the active part of the neuron, but the dendrites were not necessarily electrically active. For some time it was thought like this. Later, with the recordings, with the information from Paul Fatt, it came to be recognized that there was active impulse invasion that happens in dendrites. Also, the seminal work coming from H.T. Chang, Bishop and Clare, I think from Yale University, they showed that there was decremental conduction in cortical dendrites as the distance increased. What does this show? This shows that perhaps the information that is being conducted in dendrites are not action potentials, but rather graded potentials. Remember, it is the graded potentials that will die out as the distance increases. Action potentials will get regenerated as the distance keeps increasing. Fundamental difference between graded potentials and action potentials. In case you are not aware of this, I will share with you a video in which I have discussed what is a graded potential, what is an action potential, and the difference between the two in good detail from my previous course on neuroscience of human movement. The most convincing evidence came from the work of Per Andersen in Norway. He measured summed extracellular or field potentials in the hippocampus. The hippocampus, when I first read, I am surprised how he could have accessed the hippocampus. It was a deep tissue. So, do check how this was done. Do check out the exact methodology that was used. But what Per Andersen did observe was that there was a sharp wave that is attributed to action potentials in distal apical dendrites of pyramidal cells, a unique type of cells in the cortex, the pyramidal neurons. Do read about them because they have this special natured dendrites, apical dendrites. Do read about them. This essentially started the use of the hippocampus as a model system. This later was used to study many different phenomena, long term potentiation, long term depression, memory learning, a whole bunch of topics came to be discussed using hippocampus, but this was the starting point for all that research. Like I mentioned, so there was the population spike that was demonstrated in hippocampus by excitatory synapses, that this population spike is initiated in distal apical dendrites by excitatory synapses. This propagates through the dendrites towards the cell body and then to the axon. And the use of this to investigate trisynaptic circuits. There are equivalent trisynaptic circuits in other nuclei in the brain. For want of time, I am not discussing the details on the neurophysiology of this, but the mechanism or the first clue to the notion of memory, long term potentiation, came from the investigations of trisynaptic circuits in the hippocampus. Find out what is or what are the equivalent similar circuits that are present in other regions of the brain that are not necessarily performing the same function of memory, for example. Do check that out. Most importantly, the clearest demonstration came after the advent of patch electrode. That is the instrumentation jump or the leap that was required to clearly demonstrate forward propagation in dendrites. This was the development that was required. With this, we come to the end of this video. In this video, we looked at the active properties of dendrites, starting with both graded potentials, action potentials, and how the use of hippocampus came to be recognized as a very good model system for studying these processes. Thank you for your attention.